Now let's discuss about Human Genome Project. Children, it was launched in the year 1919 to find out complete DNA sequence of human genome by using genetic engineering technique and bioinformatics to isolate and clone the DNA segment for determining DNA sequence. And what are the goals? To identify all genes, about 20,000 to 25,000 genes in human DNA, and to determine the sequence of these 3 billion chemical base pairs that make up human DNA and store this information in database, to improve tools for data analysis, to transfer related information to other sectors, to address the legal, ethical and social issues that may arise due to project. These are the goals of Human Genome Project. And this project was coordinated by US Department of Energy and National Institute of Health. And the method involved two major approaches. The first one was identifying all the genes that expresses RNA called EST, express sequence tags. The second one, sequencing the all set of genome, the genetic pool that contain all the coding and non-coding sequence called sequence annotation. So, what are the findings? What are the salient features of human genome? Human genome contains 3,164.7 million nucleotide bases. The average gene consists of 3,000 bases, but the sizes vary greatly, with the largest non-human gene being dystrophin at 2.4 million bases. Okay. Then less than 2% of the genome codes for protein. That means 98% of the gene's functions are unknown. So repeated sequences make up very large portion of the human genome. Children, did you notice a third? That means a point C. 98% of the genes are not functioning. Okay, so in future they may be functional. We don't know what is evolution. What happens to Homo sapiens? It is we can't predict it, isn't it? Then next salient feature, repetitive sequences are stretches of DNA sequences that are repeated many times, sometimes 100 to 1,000 times. And one chromosome, that means the chromosome 1 has most genes. The chromosome number 1 has most genes, that is 2,968 genes are there. And the Y gene has the fewest, only 231. Then scientists have identified about 1.4 million locations where single base DNA differences, SNP, single nucleotide polymorphism, occur in humans. And this that is about a Human Genome Project. Children, in our revised syllabus, one more topic is related to this project, that is RISE Genome Project. So I am giving you this topic as an assignment. Okay, after watching this video today, I am not including any test paper questions. Instead, you have to do an assignment on RISE Genome Project and submit today itself. Okay, children. And the last topic is DNA fingerprinting. It is a very quick way to compare the DNA sequence of any two individuals, isn't it? Children, the lesson biotechnology principles and examples during that principles and applications. I think in the second lesson of biotechnology we had discussed this, isn't it? In the area of medicine, we had explained about DNA fingerprinting. It includes identifying differences in some specific regions in DNA sequences called repetitive DNA. Because in this region, a small stretch of DNA is repeated many times. So it is very easy to find the parents, especially in the case of paternity, in the case of murder and all. It is very easy to identify the victim. Depending upon the base composition, length of segments and number of repetitive units, satellite units may differ. Satellite DNA is classified into many categories. It is one among them. So this process polymorphism and DNA sequence is a basis for genetic mapping of human genome as well as fingerprinting. The technique of fingerprinting, now let's discuss the steps. It was initially developed by Alec Jeffrey. It was an asked question, children. He used a satellite DNA as probe to so high polymorphism was called VNTR, variable number tandem repeats. Variable number tandem repeats. Children, these are the steps. The first one, DNA extraction from the cells and high-speed refrigerated centrifuge. Then amplification of DNA, PCR, polymerized chain reaction. Then fragmentation. How can we fragment DNA by using any scissors? No, by using the molecular scissor restriction nucleases, isn't it? So by using restriction nucleases, make them into fragments, then undergo gel electrophoresis. I know these steps are very easy to use, isn't it? Because we had completed the lesson biotechnology principles. Then what is the next step after gel electrophoresis? Double-stranded DNA split into single-stranded, then southern blotting, then nylon sheet immerse. These are the steps of gel electrophoresis. Okay, so nylon sheet immerse in a bath having probes. Okay, so probes here we are using VNTR, so it is we can easily identify. Then nylon membrane pressed on X-ray film, that is autoradiography. We will connect it with the computer program. Then dark bands develop at the probe side, so we can easily identify the victim in the case of murder and all, and also in the case of paternity issues related with paternity and all. It is a good remedy, that is DNA fingerprinting. So by explaining the topic DNA fingerprinting, officially we complete the revised syllabus 2020-2021 children. Wishing you all the very best. Only my part is completed. Your part is remaining there. You have to study well, children. You have to study well. Study each and every sentence from the reader. Then only we can discuss the different levels of questions. Okay, so the concept should be clear to you. Then only we can explain application level questions, twisted questions, etc. Okay, so please do your part, children. Please study well. So next week we will start our revision. During that time we can discuss the revised pattern. A lot of changes are there in new question paper pattern. We can discuss that, and before that you complete your studies. Okay children, all the very best to you. Thank you.